students, today we'll be doing Module 35, Indicators of Food Microbial Quality and Safety. The entire lecture will be divided into introduction, the history of indicator organisms, their characteristics. Then we'll be talking about type of indicator organisms, the ones which are commonly used for testing in the food industry, their analysis. We'll be talking about aerobic plate count, yeast and mold, most probable number, and we'll be finally concluding this lecture. Now when we start talking about indicator organisms, first of all we should know what are they. They reflect the microbiological quality. These are organisms or their metabolic products whose presence in the given food at certain level may be used to assess the existing quality or to predict the shelf life of the product. This means they reflect the microbiological quality of the product. Now coming over to the history of indicator organisms. The term indicator organism was coined in 1800th century. Why? Because there were so many waterborne diseases which were being caused by the consumption of poor quality water. Then the scientists realized that we need to isolate these cultures to study them further. Carl Barth in 1880 was the first person to isolate Salmonella typhi, that is a causative agent of typhoid fever. It was followed by a major discovery where Theodore, he isolated Escherichia coli in 1885 from the fecal sources, and we came to know that E. coli is a native resident of the human gut. Once we came to know about the E. coli, then the focus was on coliform test, and gradually the coliform test was regarded as the most appropriate means of assuring the sanitary status of the drinking water supplies, and this was in 1914 by the US Public Health Services. Gradually the coliform test had the worldwide acceptance as safety and hygiene indicator. Coming to need for microbiological indicators, why do we need them? They are evidence of hazard, biological hazards. They should not be interfering with the natural microflora in the food. We should also look into the processing conditions, because some processing conditions, they remove the microorganisms and later on cross-contamination adds these to the food product. Consumers, very wide range of consumers who are using this food products, and if they are at risk, means pregnant women, children, then we have to be more careful. The state of distribution, how are they being distributed? Where are they found mostly? So we have to be careful and we have to look at their sources of contamination. At times there is temperature abuse happening which leads to the growth of these microorganisms later on. Most of these indicator organisms, they also have a spoilage potential. So we have to look into that aspect also, because they are being used as quality parameters. Ultimate consumption also affects a number of indicators in the food. Sometimes a small step like heating the food or boiling the liquid leads to a less amount of microorganisms. When we are talking about the indicator organisms, we should also be talking about the reliability of their detection. Costs and benefits, they also play a very great role, because then only we can decide that these are the microorganisms which we are going to use as the indicator microorganisms. When we talk about the indicator microorganisms, we need to know about the characteristics of an ideal indicator organism. This particular characteristics have been given by ICMSF. When we talk about the first quality which should be there, that is, they should indicate a defective process, practice, or a potential cause of spoilage. These should be stable or superior to the biological hazard or the spoilage organisms in terms of their growth, survivability, and physiological behavior. If they are not, then it's not very easy to detect them and talk about the index of quality or safety. Therefore, whenever we talk about these indicator organisms, they should be easy to detect, fast growing, and can be suitably validated, which is very, very important when we are talking about the food safety management systems. Quantitative methods must correlate well between the population and the intensity of the hazardous organism. Their detection must be easy and it should be easily implemented. All these features were focused on by International Commission on Microbiological Specification for Foods. What are these indicator organisms? These can be single microorganism or a group of microorganisms which may be pathogenic or non-pathogenic in nature. Their presence reveals a potential problem with quality, hygiene and safety of food product. Microbiological status of water and food can be estimated by talking about or by finding out the amount of these microbiological indicators. At some places, these are also known as index organisms. Why index organisms? Because their presence indicates that there is a possibility that the other pathogens or logically similar pathogens are present. Now coming to the next slide, that is commonly used indicator organisms. These can be the mesophilic aerobic bacteria, MAB. These can be yeast and molds which come from the air. We can have coagulase positive staphylococci which comes normally from the skin. Then we have the spore former, the aerobic spore formers such as Bacillus species group. Listeria is another microorganism of concern, and lastly but not the least is the family Enterobacteriaceae, the most important family which has many indicator microorganisms such as E. coli, coliforms, Salmonella, Shigella. So when we talk about the indicator microorganism, we should know their types. There are two types of indicator microorganisms. The first one are the indicators of quality. And the second category is indicators of safety. Quality indicators judge conditions which are important for product manufacturing or consumer acceptability. On the other hand, safety indicators, they identify the presence of conditions associated with increased risk of exposure to a pathogen. When we talk about indicators, we come first to the indicator of qualities. Mostly, these indicators, they reflect the shelf life of the food, which is determined by initial number of organisms present. There is no direct correlation, but these are there and we must consider the type of food we are talking. For example, raw milk will have more and pasteurized milk will have less. We should also talk about processes. High heat treatment is going to reduce these microorganisms, whereas a simple packaging will have a different effect. So when we talk about these indicators of quality, we have certain criteria for them. The first criteria is they should be present first of all and they should be detectable. The second thing is these are directly related to the product quality and we should be able to detect and distinguish them, and very important for the food industry is that they should be enumerated in a short period of time. And last, they should not be affected by the natural food flora which is present. For example, in case of fermented food where we have lots and lots of bacteria, so there should be no overlap happening with these microorganisms. Now when we talk about these indicator microorganisms, one of the most commonly used is mesophilic aerobic bacteria. When we are counting these, this count is known as APC, that is the aerobic plate count, or it is known as the TVC, that is the total viable count. What are we trying to analyze? That is the total population of bacteria in a food product, the number of bacteria that can grow in the presence of oxygen, therefore the name is aerobic, and at moderate mesophilic temperatures, which for country like ours are the room temperatures from 32 to 37 degree C. When we talk about these map, we also talk about the microbiological quality of food and we relate it to the shelf life of the products. There can be variations happening according to the type of food and processing steps it has received. The next category, which is very important, is yeast and molds. These again are the quality indicators and they have the feature that they can grow over a wide range of pH, especially low pH, acidic pH, low temperatures, even low water activity. Most of the time we find osmophilic yeast, they are growing as indicators or they are growing in low water activity. Foods like jams, syrups and juice concentrates, where they can be used as indicators of quality. For example, Geotrichum candidum, the machinery mold, acts as an indicator of plant and equipment sanitation. If the sanitation is good, the chances of finding Geotrichum are much less if there is no control on the hygienic cleaning of the plant. Now when we talk about the quality indicators, they can be for cereal grains and their products. They can be yeast and molds, which can range from 10 raised to the power 2 to 10 raised to the power 4 CFU per gram. We can also have alcohol tolerant fungi and bacteria. These are indicator of spoilage of wine. Then we can have Leuconostoc mesenteroides, which is seen in the sugar refining industry. On the other hand, we have lactic acid bacteria and the flat sour spores. We also have Acetobacter species in beer, wine, canned vegetables, fresh cedar respectively. We are coming over to the next group of microorganisms. They are Bacillus cereus. It's a very important group because these bacteria are ubiquitous in nature. They are found everywhere. They come from the soil and usually they are the indicators for raw material quality as they are found almost in all foods. They can be used as a marker to find out the efficiency of cleaning processes. They have a very strong capacity to adhesion to stainless steel surfaces accumulating on the manufacturing equipment. They are also there when insufficient thermal treatment is given during a particular process. They are also found during the storage at inappropriate temperatures that allow their growth and after the thermal treatment. We can find Bacillus cereus group in rice, potatoes, pasta, milk powder, etc. The next group of microorganism is coagulase positive staphylococci. These are used an indicator of good handling procedures and they also indicate contamination from the workers because they are found on the dirty hands, skin, nose, and they also come through the throat infections of the worker. When we talk about the next group, Listeria, although people feel that it's an emerging pathogen of concern, but it has really established itself as a pathogen and it is a gram-positive rod which is able to grow at refrigerated temperature. Therefore, it is used as an indicator for the potential presence of especially Listeria monocytogenes species during the refrigerated conditions. It is also taken for the various food processing environments to predict the cleanliness of the equipments. When we talk about the types of indicators, the next type is the safety indicators, and these indicators are very commonly used in the food industry. These indicators denote increased risk of pathogen contamination in foods and water. So when we talk about these CFT indicators, the most important group here is the coliforms, which indicate about the water quality. We have certain fecal E. coli forms which are there and they indicate the possible contamination with enteropathogens. Why are we talking about enteropathogens? Because they come as a source or they come along with the fecal material, and therefore there are chances of finding other pathogens. When we talk about the coliforms, these can also be used as an indicator of sanitary production and post-processing contamination. So when we are talking about the indicators for safety, we should also know the criteria. Criteria more or less are very same with the quality indicators. The first and the foremost is that they should be easily and rapidly detectable. We should be able to distinguish them from one another. We should be able to differentiate when a pathogen is present. We should be also able to talk about their numbers, their growth, and their death should correlate to the pathogens which are absent from foods. When we are talking about the foods, the coliform should indicate that the pathogens are there or not. Family Enterobacteriaceae. It's a very important family when we talk about food safety indicators. This particular family can be used as a tool to assess the adequacy of processing and hygiene practices, mainly in heat-treated foods. Most of the members of this family are killed by the thermal processes applied in food production, and their presence in processed food can be considered as an indicator of insufficient, faulty processing or post-processing contamination. A major important group of family Enterobacteriaceae are the coliforms. These are gram-negative, non-spore forming rods that ferment lactose to acid and gas. This formation takes place within 48 hours of incubation at 37 degrees Celsius. Like I said, coliforms, they belong to family Enterobacteriaceae, and there are many important genera. Why these genera are important to us? Because majority of them are foodborne pathogens. We can have genera like Citrobacter, Enterobacter, Escherichia, Klebsiella, Salmonella, Shigella. All these microorganisms are pathogenic and they pose a public health threat. When we talk about coliforms, there is an important group, that is the fecal coliforms. These have the same features as the other E. coli. They produce acid and gas, but they grow at a temperature of 44 to 45.4 degrees Celsius. They act as indicator of fecal contamination, because if these E. coli are present, then the chances are very likely that the contamination has taken place from a fecal source. We have been talking about the indicator microorganisms. A very important part for these indicator microorganisms is that we should be able to do their analysis. When we talk about the analysis of indicator microorganisms, there are various types of methods which are being used. The most common is the APC, that is the aerobic plate count. It is also known as standard plate count or SPC. We use Newton agar or plate count agar for this particular method. Aerobic plate count is a conventional method and it takes lots of time, and we get the results after 24 hours. Therefore, people have been working on the rapid methods to find out or to enumerate these microorganisms, and these rapid methods can be the membrane filtration technique. We can use spread or pour plates, and then we are incubating these at 37 degrees C for 12 to 18 hours. This is one segment of microbiological analysis where automation keeps happening so that we can get faster results. Then we talk about yeast and molds. The most common media which is used for their growth is potato dextrose agar, that is PDA. Since mold colonies are able to spread very fast, we are also using selective agents such as chlororamphenicol, rose bengal dye and dichloron. Most of the plates are spread or pour plates which are incubated at 25 degrees C for 3 to 7 days. When we talk about the family Enterobacteriaceae, and we have just seen how important or how significant is this particular family. It comprises of coliforms of fecal origin and Escherichia coli. We have a wide variety of media which is being used, such as the violet red bile lactose agar. We also have the lauryl sulfate tryptose agar. We have the brilliant green bile lactose broth. We can also use eosin methylene blue agar. And to differentiate between the members of family Enterobacteriaceae, we can also use a battery of biochemical tests which is known as the IMVIC test. These are indole, methyl red, Voges Proskauer and citrate utilization. This test are able to differentiate between Salmonella, E. coli, Enterobacter, Shigella. Last but not the least is most probable number, which is commonly used across the entire water and food industry. This is a very statistical based method, but based on probability. It is based on the growth patterns of positive and negative tubes. We are able to select the tubes. The positive tube means that there has been a change in the color and there is a production of the gas. These positive tubes are then counted with the help of the table which has been given by the McCarty, and it is known as the MPN table. Once we get the MPN value, then we can talk about the probability of these coliforms in the water, and we can go ahead with the presumptive or the completed test in case of coliforms. In the end, I would like to summarize this lecture by saying that indicator organisms are very important for quality and safety of water and food. They find a special reference in all the regulatory guidelines across the globe. They are very effective indicators for various types of food applications, but we cannot take both the indicators to achieve all the goals, that is safety and quality, together. Indicators must be carefully chosen and they have to be used in a proper situation, and we have to also consider the product processing conditions, the metrics of food where we are talking about their presence. The methods, they should be simple, cost effective, they should be easily, and the techniques have to be very rapid, because then only we can have efficient estimation and detection of indicator organisms. Thank you.